Hello, and welcome to this week's episode of I Came for Science, a show about sex and reproduction and inclusion in STEM. I'm your host, Laura Surratt. I'm a professor of biology, and I've been studying animal sex for the past 30 years. Please join me as we normalize talking about sex and what we know about it, as well as the journeys of the scientists who study it. In today's episode, we'll be taking a deep dive into a process called sperm storage, which occurs when sperm viability is maintained after leaving the male reproductive tract. We will first explore how this works in non-human animals, and then learn about how humans are able to store sperm and maintain their viability for decades, and why people might do this. I'm so pleased to be joined today by my friend and colleague, Dr. Teri Orr, who is an assistant professor of biology at New Mexico State University and co-author of a book entitled Reproduction in Mammals, the Female Perspective. So Dr. Orr will tell us about her research, her journey through STEM, and also about sperm storage in different non-human animals. Welcome, Dr. Orr.
Thank you. I'm excited to be here.
We are so excited to have you. Can you please start by telling us about what area of reproductive biology you study?
I'm really interested in how female mammals make decisions in the context of when they might have their offspring. And there's a physiology we call reproductive delays, and that includes sperm storage. So it's anything that a female might do that can change the timing and usually make it later rather than earlier for her to be able to have these longer periods of time. So she might mate and then store sperm. She might mate and then not store sperm, but then do something we call delayed implantation. So that's where essentially that developing blastocyst, which is a ball of cells that has genes both from mom and dad, it doesn't implant. So I study mammals. So these are animals that usually have implantation and eventually a placenta. So it won't implant for a few months. And we see that in bears and walruses, seals, some skunks, a few species of bats. And then the other type of delay is called delayed development. Sometimes it's also called embryonic diapause. It's not very common, but it happens when an embryo has started to develop. So you have a placenta, but then there's no more cell division. And that can last for a few months, where essentially that little embryo is hanging out and there's no division of cells. It's just staying the same size. And that can extend the time over which that mom might give birth by a few months. So she'll give birth a couple months later than she would if she didn't have that. So I'm really interested in why do all these females, different species have these? And then once you have one of these types of delays, how does that impact other parts of your biology? And so one area I'm really fascinated, which I think is how we met, is through sexual selection. And so where males and females may interact through different pressures for the two of them to have their interests kind of played out.
Mm-hmm.
But it may have immunological consequences, right? So if you've got this foreign body, which a baby is developing inside you, you have to regulate so that you're protecting yourself from infection, but you're also not attacking that developing offspring. So those are kind of the things that my lab studies. We usually study them in bats because bats have all three types of delays, whereas most only have one or the other. Yeah.
Very cool. So the sperm storage, the delayed implantation, and the embryonic diapause, are those the three?
Exactly.
Yeah. Very cool. So how the heck did you get into this area of research?
I would say in terms of bats and delays, it goes back to being an undergraduate in a mammalogy class. I went to the University of New Mexico and I was sitting in a mammalogy class, and the instructor was telling us about how some species of bats, when they're hibernating, the females will be torpid or hibernating. The males will come out of torpor or hibernation, and they'll go around and they'll look for females and they'll mate with them. And then the females will store sperm. So the females are essentially out. Okay, sleep and hibernation are two different things, but certainly when you're hibernating, you aren't kind of like controlling what's happening, right? So they're basically have these males mate with them, and then 4 months later, then fertilization, which I like to call conception. We can talk about why I use that word instead, but conception will happen, and then pregnancy will occur. Well, bats, they only have 1 or 2 babies every year, and they take a few years to even start reproducing. so it's really expensive for them to reproduce. So the fact that these females are not seemingly exerting any choice about who the male is was really problematic for me. And the instructor, who was a great professor, et cetera, but it was kind of just a guy like, oh yeah, well that's just how it happens. And I was like, no, 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 no. There's got to be more to this than just the females hibernating and then whoever the heck can be the father of her very, very precious young that she only gets to have a few of in her life.
Right. Oh my gosh, I have so many questions. Okay. So first of all, how many males does a female mate with?
Yeah, so that's a great question. So because bats usually only have one offspring per litter, I'm going to call it a litter,
Sure. Yeah.
per reproductive event, we can't always do paternity testing to reveal
Oh,
that
to
answer, right?
know. Right.
There's only one dad because there's only one kid.
Right.
However, there are some species that have twins or triplets, and
Uh,
from those we know there can be multiple dads.
so interesting. So you're not actually watching to see which males mate with a female.
Not yet. That is
Okay.
certainly something that off and on I've wanted to do. It's a little bit more logistically tricky
Yes.
than one might think because all this happens at night and
Right.
they're
Oh
very
my.
cryptic.
Okay. So you were like, this doesn't add up. Like,
This doesn't add up.
doesn't make sense. The way it was being taught was like the females just accept being mated with, and whose ever sperm they happen to get are the ones they use. And you're like, something else must be going on here.
Yep.
Interesting. So let's put a pin in that and we'll come back to that. So going back even earlier than that, you were in the science class, but how did you even get interested in studying science in the first place or in the natural world?
Yeah, so it's kind of interesting because I'm first generation, so I didn't have anybody in my family other than some brothers and sisters that had gone to college.
Mhm.
But I was really curious. And so I was always asking questions, always observing things. And, I from movies, I'm of the age that, Indiana Jones was like the hotness, right? Like, oh,
Right.
I wanted to be an archaeologist or
Uh-huh.
maybe a paleontologist. I thought that's cool. And you may or may not remember, but he was a professor, right?
Mhm.
And then would go off and have these adventures. I'm like, well, that seems like, roughly what I want to do. So I knew I wanted to be a professor, but I mean, this is kind of embarrassing now, but I didn't even know that meant going to grad school. Like, I didn't know
Mhm.
what grad school was. I
I
just
don't
knew I
think that's
wanted to like
embarrassing. Like a lot of people don't know. Yeah.
yeah, yeah. So I was like, well, I'd love to teach and I'd love to like, go out in the field and do things. So that was kind of where it started. I ended up, going to the University of New Mexico for archaeology. But what's
Okay.
funny is as a kid, my dad was an artist and he used all, natural things in his artwork.
Cool.
So feathers and leather and, plants and what have you. My grandmother was a really avid gardener. And so I had those influences and I loved nature. I loved animals. So I never put two and two together. I just thought,
Right.
well, study archaeology, do that. Once I got to, my very, very first semester, I applied to a biology class, or not applied to, I registered for a biology class and really fell in love with it pretty early on and realized that I already had a lot of knowledge from just growing up the way I did. You kind of think, well, I don't know anything about anything. But then you realize, well, actually, I do. Like, I know the difference between an annual and a perennial, and I know
Mhm.
what this bird is and that bird is because of how I had been raised. And
Yeah.
so I really quickly realized that biology was a much better fit for me. I finished with a dual major, but yeah, that's kind of how that happened.
Were there other really important points in your academic journey that you felt like you were either encouraged or discouraged from pursuing advanced studies or a career in STEM?
Yeah, I would say certainly it's about that time, when I was in college, where when I would go home and I would visit with family friends and that sort of thing, they couldn't understand why I was even going to college to study mammals and,
Mhm.
why was I doing what I was doing? And so the question I would get was like, oh, well, you're doing all this field work. Is it so that you can be an exterminator? That was like the
Mm,
only practical application they could
right.
come up with, right? So it was hard to kind of extend or relate that material back, especially early on. My family was super encouraging and proud, which was really, really nice. And so I always felt like I had that support, which was awesome. And during my first postdoc, I think this is
Mhm.
kind of remarkable. My postdoc mentor, Patty Brennan, from her name you would never guess, but she's Colombian
Mhm.
and not a native English speaker. And she's a really great writer. And she went out of her way. I remember she would work work through my writing with me. And she said, look, you
Wow.
can do this. You can
Mm-hmm.
do this. And it's going to be a lot of work. And
Mm-hmm.
you may always have to have people editing your work, but this is possible. She's like, you will get better. And that support was massive for me.
Like really believing in you, having a growth mindset, not you're just bad at writing and you're going to stay there, but and
Yep.
here's how I'm going to help you. That's
Yep.
awesome. Related to that, we've talked about some pieces of this already, but are there any other aspects of your identity or lived experience that influences your science?
I mean, I think one thing that comes to mind is, being from kind of this rural area and as a woman, I definitely was kind of raised as like, you go help with the dishes, like
Mm-hmm.
you're quiet and meek and you're not meant to, kind of that sort of attitude of women are meant to kind of, I mean, I was always encouraged to be curious and do my best, but also told, that I should basically be doing these kind of feminine tasks or,
Mm-hmm.
exude this kind of feminine nature. I don't know how to phrase that better. And so I think that dichotomy of like you can do anything / you should be quiet and help with the dishes and
Mm-hmm.
kind of made me feel, I don't want to say confused inside, but it did make me kind of question like what was actually right, what was actually the way that things ought to be. And so going back to my story about the bat and how these females were just there and the males were around and the females were kind of
Right.
passive, I think it kind of has made me be a little bit more questioning about those cases where we have these assumptions that aren't necessarily correct. They aren't necessarily what's going on.
What's actually going on? That's a beautiful tie-in. Great. Well, I'm sure our listeners are just dying to hear about your research some more. So you kind of gave us some tantalizing bits about the females being in torpor and the males mating with them. So take us further into that story. So what is actually happening?
Well, there's a lot going on. And so I also want to say, we talk about something called cryptic female choice. That's not the only type of sexual selection, but I want to start there just to mention.
Sure.
So I think
Yeah. And
that's,
distinguishing between conception and fertilization and all those
yeah,
terms. Yeah.
exactly. So
So
yeah,
let's define some terms here.
yeah,
Yes.
let's define some terms because I do think that clear thinking helps us kind of like clearly establish what we're talking about. So cryptic female choice is also something that I find really fascinating. And the idea is that it is anything that the female might do that's within the confines of her reproductive tract. So that's in the uterus, the vagina, the oviducts, all that. Anything she might do that biases who the dad is going to be, we might call cryptic female choice. And it's called cryptic female choice because you can't see what's happening. The word choice doesn't necessarily mean that it's conscious, right? So it could just be through the nervous system doing things, etc. So we don't want to, assume that the choice is an active choice. I want to be very clear about that. Let me just clarify when I say conception versus fertilization. So fertilization is something that a sperm is doing, right? And so it's male active, whereas conception, I like because it's totally neutral between the two sexes of who's doing what. Conception just means that two gametes have fused together, and now you've got a thing called a conceptus that is going to go on to be potentially an offspring if it, does well.
And then there's sort of the sperm movement from the male into the female. And what do you call that?
Insemination. That's a great point. Yeah. So
Yes.
insemination. So again, it is kind of a
It's
male
a male
active.
active, right?
Yeah.
Yeah. And sometimes there's like some species where the males will deposit a spermatophore and the female has to actively pick it up. So,
Yep.
yeah. So it's sort of like female receipt of sperm, one could say. Yes. So when the female receives the sperm, which can happen in different ways too.
Exactly.
Interesting. So that gets us into this idea of sperm storage too, that you have the conception, but that's not necessarily at the same time as when the female receives the sperm,
Mm-hmm.
right? Do you want to take us down that track a little bit?
Sure. Yeah. So, it's really interesting. I've argued that basically all species that have internal insemination or sperm receipt or whatever we're going to call it,
Yeah.
that they store sperm for a slight period of time in the sense that, the ovum isn't just hanging out right there waiting, and the sperm, conception doesn't happen immediately. It might take some time for the two gametes to meet each other. So any species where fertilization or conception is happening internally, you could say that there's some form of sperm storage. But in terms of, these more extreme cases, so I will say in humans, this period of time that I'm talking about, where I say anybody who has internal conception has some form of sperm storage, it could be anywhere from a few days up to almost a week that sperm might be viable.
Remember that, folks.
I think
You
that's a
leave with
good
nothing else.
life tips, right?
Mm-hmm.
Yeah, so up to a week. Whereas, there are some birds that it can be a few months, and then in bats it can be up to a year in some species. The winner, and I think this is amazing. Actually, I'm going to tell you a weird one first. So, earthworms, and I know earthworms have all kinds of crazy things going on, but they can also store sperm, and they can do it for a couple years. And I didn't
know
even know
that.
they lived that long.
Right. Wow. And they're hermaphroditic too,
Yeah, and
right?
they're
Yeah.
hermaphroditic, but that kind of, in earthworms, and there's other cool hermaphrodites where that just means, that they might produce both ova and sperm, but they're used in a similar fashion.
Right.
So
But so they're storing sperm from another earthworm
exactly,
for
yep.
a year. Okay.
But then the other really cool one are leafcutter ants. So a female leafcutter ant, she'll mate with a male, and then she stores sperm for the duration of her life. And for these guys, it can be about 10 years that they're storing the sperm, which is
Millions
really
of sperm and keeping them alive. So this is the fascinating thing that I wanted the listeners to hear about. It's wild to me when I think about it. I mean, I know it, I study it, but that these cells from another organism can be maintained alive and able to conceive with or fuse with another gamete for years. That's wild.
Yeah, so one of the things that is really interesting is going off of what you just said. You've got this cell that's now in a foreign body, so it has to evade the immune system or be somewhere where it's going to be protected from the immune system. Because if you think of, we can think of ourselves, if you had a infection within your reproductive tract, you would want to clear that out, right? So there's any foreign substance that comes through, and we know that like the vagina is really acidic, and there are all these things that we have to keep our reproductive tract healthy. And so those sperm have to be able to evade that, but they also have to be able to live for that whole duration. But what's really wild is in most cases, especially the mammals that have sperm storage, we don't see that the female is clearly providing those sperm with anything. So the single cell has to have with it sufficient nutrients or energy or whatever you want to call it to be able to last that whole period of time. As much as that's like, wow, that's fascinating, we should know more, we don't. We don't. Other than a few really case studies in things like fruit flies. I would say maybe mosquitoes, right? We don't really know.
Yeah, not even that well in mosquitoes, but fruit flies, the kind of classic genetic model of Drosophila melanogaster. We're learning more and more about what proteins are important for sperm to get into storage and then to be released from storage. And this is mostly from the lab of Mariana Wolfner and Yael Heifetz, and really finding interesting neurological, muscular, and biochemical interactions between males and females that are involved. And then some appearance, but I don't think we still really know like what's actually maintaining them metabolically.
Yeah, so all of that has been really fascinating for me. So one study I did that ties in a little bit more with the male side, and I do want to point out, I study both sexes because both are interacting through all these processes. And so one thing that I did with my first postdoc mentor, who I've already mentioned, Patty Brennan, we actually looked across bat species that have these different types of delays, many of whom have sperm storage. We examined the genitalia because the males have these crazy spines on the genitalia. This has been seen in other taxa too, like seed beetles. There's some beautiful work from Sweden. Goran Almqvist has done some work with this, where they've actually even been able to remove these spines that the males have on their genitalia.
Yeah.
See what happens. But in the bats, they've just been really poorly described. And so what we've done is tried to look at whether or not in species where the female stores sperm, the males in those species also have spines. So not all species have sperm storage, but also not all species have these spines. So we've kind of thought there might be a correlation between the occurrence of these two traits, where essentially the females who store sperm, the males of those same species might be using these spines to remove the sperm from their competitors, so from other males, before depositing their own sperm.
Gotcha. And that happens in the damselflies that I used to study. Yeah. Yeah. That males have all sorts of these scoops and spines and swirls on their penises that appear to play a role in removing other male sperm from female sperm storage. Yeah. It's so wild.
Really wild.
So has
Yeah.
that been shown in any mammals before this?
No, it really hadn't been looked at, which is
Interesting.
surprising to me. And we did, so the punchline is we did find an effect. So it does look like there is a co-occurrence of those two traits.
Where do females store sperm in bats?
Oh, that's a great question. So part of the reason bats are so cool, there's so many reasons, but
So many
one of the
reasons.
they store sperm without any specialized structure. So yeah, if you were to look at any of the insects that you've studied,
Yeah.
I'm seeing all the cool structures they've got, and
Mhm.
sharks, and I'm trying to think of other examples. Birds, they have sperm storage
Those
tubules.
tubules. Yeah.
But bats, they just use the pre-existing structure. So
Hmm.
they store sperm at that junction between the oviducts. Some people call oviducts fallopian tubes. I don't use the word fallopian tube because that's named after a dude. I like terms that are more anatomical. So
Mhm.
the oviduct is the duct upon which the ova, they go across
Yeah.
it. So right at that junction, there are often sites where females will store sperm or folds within the uterus. Some people say that dogs store sperm. And again, like I said, I think anything that has internal conception has some period of sperm storage. Well, they have uterine glands, just like we do, tiny little glands in the uterus, and the sperm will get their heads in there and kind of hang out in those little
Oh.
areas. So what is interesting is when these sperm embed in the female's reproductive tract, it's
Mhm.
rather deep. The
Oh.
whole head is embedded within her tissue. So to me, if you look at microscopy, so images under a microscope, if you look at those, you'll definitely say, wow, those are like really invasive, like they're really in there. And the ones that aren't embedded seem to get sloughed off. So that could just be protecting them from the immune system, from
Mhm.
the flow of fluid around them. But I think more likely than not, that suggests that there may be the opportunity for some sort of nutrient exchange. The other
Mm.
interesting thing about bats, and so bats have the longest period of female sperm storage of any mammal, up
And
to a
how
year.
long is that again?
Up to a year,
Up to
which
a
is,
year. Wow. Yeah.
yeah, that's probably on the high end, but
Right.
many species will do it for a few months.
Mhm.
And if you look at the midpiece, which is the part of the sperm where they have a bunch of mitochondria, and mitochondria are the powerhouse of the cell. That's where we get ATP, which is the energy that we use. For bat species where they have sperm storage, female sperm storage, you'll see that those midpieces are really long.
Hmm.
The males may bring their own machinery with them,
Mm.
the
Mhm.
situation, right?
Mhm.
And what's interesting too is with female bats, we also know, so going back to your question, they will expunge, they will eject the sperm from some males. And so
Mm.
remember those hibernating bats? So even though they're hibernating, what people have noticed, which is really interesting, is that, if you had a video camera on them, a thermal camera on them, you'll actually see them. First and foremost, if anybody's interested in seeing this, Attenborough has some footage in the
Okay.
life of mammals where you
Okay.
can see that the females are hibernating, they're cold, they're dark, because
Mhm.
that's how that imaging works. And the males will come out of hibernation, they get warm, so they get red, and you can see them go to the females, mate with them, and they'll transfer that heat while they're doing their business. And then you'll see that the female is still hibernating after, because she immediately, at once that heat is kind of away because the male has left, she's
Mhm.
back to cool. But the females will actually expunge or eject the sperm from that mating sometimes.
How do
So
you know that?
people have observed the little blobs falling. Yeah, falling
Interesting.
out. The other
Wow.
thing, the other part of this story, there's actually two other parts. So one is that there's something called a copulatory plug that I haven't mentioned.
Mhm.
A lot of different species have them. So, they're seen not just in mammals. I want to be clear
Mhm.
about that. But in
Yeah.
the bat, it's kind of like a little blockage of the vagina. And the interesting thing is that you can envision that it could have different origins, right? It could come from the male, where the idea is that he's trying to prevent other males from mating with that particular particular female so that his sperm will get used, again, sperm competition, right? But it could also be from the female, and it could be her trying to prevent remating. It could be a crosstalk between the two. What we do know is that males will remove those plugs. You'll actually see them biting at it, removing it, and throwing it on the ground, and then mating with her. She will also remove them.
While she's in hibernation?
Not all,
Okay.
I point out, not all species that have sperm storage hibernate.
Okay, gotcha.
And so we'll jump back a little bit and
Sure.
talk about, the ones that don't. And so the ones that don't, we'll see that she will remove these.
So these are active matings where the female's awake, actively mating. What about those females who are in hibernation? Do you know anything now about the mechanisms by which they might be influencing whose sperm are fertilizing their offspring or who they conceive with?
No, not really. Not yet. That's the answer. Exactly. Not
Yes.
yet. So that's still a bit of a mystery that we're trying to sort out. Certainly, we know that these stored sperm do lead to viable conception. So we know that they are used, which is really an important piece. It seems like a duh, but that's because we know it. We didn't always know that. We would see these matings, and people for a long time assumed that those sperm were being used, but she just as well could mate very quickly when she comes out of hibernation, and then those are actually the sperm that are used.
Yeah.
So we do know that they are used. We do know that they can come from multiple males, so that maybe she's using sperm from different males. But beyond that, we don't really understand the mechanisms. The other thing that I do want to mention, because I think it makes the story even crazier, is that we've been talking about the females. And again, as much as I do try to take a female-centric perspective, I try to always remember to study both sexes. And the males of most of the species that have female sperm storage with this hibernation aspect that I mentioned, they also store sperm.
What?
Well, yeah, so we've been ignoring them, but now I want to bring us back to that because it's really important. So in the summertime, the males will undergo spermatogenesis. That just means making of sperm. And then they will take those sperm and they store them in their epididymis, and they will store them there for a few months. They will store them there through the fall and the winter until they go and find those hibernating females and mate with them. Or in spring, they might mate again too. There are some matings that happen once everybody comes out of hibernation as well. And the other cool thing that we've done, and I have a student that helped with this project, an undergrad, Jennifer Silva. Basically, the males of some species have this really dark pigment around the epididymis. It's almost coal black. And so there is a really neat paper, because marsupials, for some males, they'll have a little bit longer lived sperm than others. And so there was kind of evidence, but nothing, hard and fast, that maybe it helps protect those sperm from some sort of UV damage. And so with Jennifer, what we did is we actually looked across all bat species that have sperm storage. And again, there's bats that don't have sperm storage, and there are bats that have other kinds of delays. So we identified who had what, and then we went to museum collections, and she basically determined whether or not they had this melanization. And we found really strong evidence that in the species where males store sperm, they also have this black protective layer that keeps those sperm. I mean, the idea, it's indirect, right? The idea is that they're protecting from UV. We looked at other things like, where are they roosting? Are they exposed to light? How much light might they be exposed to? So there's some other little side ideas that go with this. It is interesting because we think of them as being nocturnal, and why would they ever get light? But of course, some of these species, they roost under leaves, they roost under tree bark, so they get a little bit of light. But we do see this relationship with this melanization.
So interesting. How close is the epididymis to the surface of the skin?
Really close. So bats are cool because they're so precarious, energetically speaking. They don't carry a lot of fat on them because it's so costly to fly. You can see, the epididymis. You can see that it's full and they're storing sperm or not. So everything's just right up the skin.
So you don't even have to dissect them to see that.
No, you don't.
Oh
Yeah,
my
that's
gosh.
the cool thing.
So what other cool stories are there out there of sperm storage in animals that you'd like to share with our audience?
One that comes to mind is that in some species of spiders, the females have different spermatotheca, the storage organs that we've been talking about. And when they mate with males, they are able to partition the sperm from different males. So male A, B, or C might go into spermatotheca A, B, and C. This is work that comes out of Mediane Adrade's lab. So she's been able to show this. And I think it really highlights the fact that females females have the capability to use these structures in really interesting ways. Now, again, I'll highlight this isn't necessarily a conscious decision, but it's something that is happening physiologically.
Such an interesting story. This has been such a fascinating conversation. Thank you so much for joining us.
Thanks so much. This has been great.
Now we're going to turn from talking about sperm storage in non-human animals to talking about sperm storage in humans, which is a fascinating topic. And I'm pleased to be joined by Matthew Hudnall, who's the Director of Lab Operations at CryoBio, which is an international sperm bank, and at Reproductive Diagnostics, which is a fertility preservation center, both located in Columbus, Ohio. In addition, Matthew Hudnall is the President-elect of the College of Reproductive Biology. Thank you so much for joining us.
It's my absolute pleasure to be here. Thank you for having me.
I have so many questions about sperm storage in humans. It's such a fascinating area. So first, can you tell us a little about what your job entails?
Yeah, sure. So primarily my job entails the oversight of operations at both laboratories for the Sperm Bank and the Fertility Preservation Center. And this involves the management of all the samples that we store, as well as compliance with regulatory associations such as the College of American Pathologists, the FDA, State of New York, CLIA, Ohio State Department of Health, so on and so forth. So really it's oversight of daily operations, quality control, cryo inventory management, and regulatory affairs.
Wow. So this is an interesting line of work. How did you get into this area of science?
Yeah, it's not something that I ever saw myself getting into. It was happenstance that I got introduced to the field. So for some period of time, I was doing graduate studies at Kent State University here in Kent, Ohio, in biological anthropology, and under C.O. and Lovejoy, who you might be familiar with, National Academy of Science member. And my interest in reproduction was really sparked then based on the evolutionary aspects and the different reproductive strategies of the great apes, how humans differ from chimpanzees, differ from gorillas, and how these reproductive strategies affected not only the behavior, but also the physiology and the anatomy of these animals, and how humans took over the planet. And that's really got my interest in reproduction. And from there, I started doing some lab work and found an IVF center that took a chance on me and allowed me to get introduced to IVF, which is in vitro fertilization, on the weekends in their laboratory. And then what that evolved into, I was diligent, I proved my ability. So then I was offered a full-time position there, working with sperm samples, which then eventually led me into also working in embryology, dealing with the IVF aspects of things, and endocrinology, performing all the blood tests for the hormones, and then managing those laboratories at 5 different locations around the state of Ohio. And that's really where I got my start. And I spent about 11 or 12 years at this center before moving into my current position. And also, I think we are all personally touched in our lives at some point by people in our lives, family, friends with cancer diagnoses. And it was from early on in my career fascinating to me how cancer actually affects reproductive health, the disease process itself, in addition to the treatments for the disease. So I really became interested and involved in fertility preservation for cancer patients. And that has also driven me. So I think personally, both my outlook on access to reproductive health, as well as lived experiences with cancer diagnoses, that has shaped my pathway in this field.
That's really important. Thank you for sharing that.
Yes, absolutely.
So you brought up cancer patients. I was wondering if you could tell me about what different types of people seek your services at the International Sperm Bank and the Fertility Preservation Center.
Sure, absolutely. Great topic. So as far as CryoBio, the sperm bank, is concerned, we provide donor sperm services to patients. So what leads people to seek our services here, many different instances. So I think for one, of course, heterosexual couples to which the male has some infertility issues and they have to go the donor sperm route. And that's the classic example of who seeks out donor sperm. In addition to that, now also there are same-sex marriages of female-female couples who seek to build families. And in order to do that, obviously it takes a sperm. So we offer services for those patients as well. And there's a couple different pathways to parenthood for those patients to take. One, of course, is choosing an anonymous donor from the CryoBio catalog of donors, one that suits what they are looking for. The other option, which a lot of people choose, is what's called a directed donor. And what a directed donor is somebody who is important in your life. So it's a known donor, someone that you know. So they go through the entire same FDA regulated process that an anonymous sperm donor goes through, but their sperm then is only used in the treatment for the couple that sought to use their samples.
Gotcha.
So that's one. So we covered the heterosexual couples with male infertility, and then the same-sex couples. And another demographic that we serve is single by choice mothers. Women who want to have children, but are not in a marriage or do not have a partner and do not seek to have a partner. Then we provide them services as well. Now, as far as the Fertility Preservation Center is concerned, there's a lot of different demographics of people that we can assist with fertility preservation. So for one, we store embryos, eggs, and sperm for individuals as well as for IVF centers, because a lot of IVF centers don't have the room in their facilities to house samples.
Interesting. I never thought about
for
that.
patients.
Yeah.
If IVF practice, if they've been in practice for 20 years, to preserve all of those samples takes a lot of room.
Right.
And the clinics don't have that room. So that's where we come in. We also provide fertility preservation where we freeze samples, semen samples on site. And this is done for a multitude of reasons. First of all, after the overturn of Roe v. Wade, we saw a spike in men who were seeking to have vasectomies. The problem, however, lies in the fact that maybe you will want a child in the future, even though you don't want one right now. And, that changes over the course of time. We might say we know adamantly, 100% right now, I never want kids. But 10 years down the line, you might change your mind. So it's always a good idea before you get a vasectomy to freeze a semen sample. That way you have preserved your fertility on down the line, and you can be used to establish a pregnancy and a partner later. We talked about cancer patients. So before any cancer treatments start, a lot of cancer treatments are gonatotoxic. And what I mean by that is that a lot of the treatments can affect the function of the gonads, whether testicles or ovaries, and their capacity to produce sex cells.
Can we pause there? Can you explain why that might be?
Yeah, I think, a lot of the chemotherapies, let's say the heavy metal ones like cisplatin, that's based in platinum, it's just heavy metal toxicity on the tissues that then disrupts spermatogenesis or oogenesis, those processes. And then the radiation treatments just harm the tissues, cause other mutations that just make the process difficult. So fertility preservation prior to cancer treatment, fertility preservation prior to a vasectomy, and also fertility preservation prior to gender affirmation treatments. So whether you undergo hormone replacement or whether you take the surgical option, again, you may say right now, I don't want kids, but so many times that changes in the future. So prior to the administration of hormones or prior to the affirmation surgery, we also always advise for fertility preservation in those instances.
Which leads me to my burning question about this. So earlier in the episode, I talked to Dr. Orr about how non-human animals store sperm once it leaves the male, but how the heck do you do that with humans? And what's the science behind sperm preservation?
Yeah, sure, certainly. So first, of course, a sample must be produced.
Yes.
Once that sample is produced, we run our analyses on it, which include a count of the sperm concentration, how many of them are motile, the motility, How many of them are moving in the direction that they should be moving and not just swimming in circles, right?
It happens.
Yes, it
Circles
does.
happens.
It does. And various other parameters. There's more than a handful of parameters that we analyze. So we analyze those parameters on the neat sample, on the raw sample itself. And then we introduce to the sample what are called cryoprotectants. Now, the cryoprotectants permeate the cell membranes. It basically creates an osmotic gradient. And when we're freezing
Can we define osmotic for our audience?
Yeah, sure. So high to low salt concentrations is really what we're concerned with here. And introducing those too quickly will, of course, harm the cells. So this has to be done very slowly and very methodically. Now, the reason we are doing this is to displace the water from inside of the cells. When we freeze these cells, we are freezing them, as I stated earlier, in liquid nitrogen, which is negative 196 degrees Celsius. And that's about negative 320 degrees Fahrenheit.
Wow.
So at that temperature, all metabolic processes cease and the cells are preserved in time. But in order to do that, we need to prevent the formation of ice crystals. Because if ice crystals form, then of course we know they're all jagged and sharp, and they can lyse the cell membranes and kill the cells. So in order to freeze these samples and then later thaw these samples and recover live cells, we need to prevent ice formation. So we do that with these cryoprotectants. Now, these cryoprotectants, the main one used in sperm cryopreservation is what's called a test yolk buffer. And what its composition is glycerol. And then also, hence the name test yolk buffer, actually egg yolk is used as media from chickens, yes, from FDA cleared virus-free chicken stock, as well as gentamicin, just to control any kind of microbial activity that may be.
It's an antibiotic.
It's an antibiotic. Yes. So glycerol, yolk, and gentamicin are used and, as I said, introduced slowly into the sample just to displace that intracellular water, which actually shrinks the sperm. So the sperm actually, they're already the smallest cell in the body, as opposed to the ovum, the human egg, which is the largest cell in the body, which is kind of this neat little dichotomy that exists in human biology. But yeah, so we displace that water. And then also that media that we're using never fully solidifies. So it kind of stays in a liquid state. So we've displaced water from inside of the cell. So there's no ice crystal formation. And then around the cell kind of stays in a semi-liquid state while frozen at negative 196 C or negative 320 Fahrenheit. And that way we prevent ice crystal formation. But like I said, that methodology has been around for a couple of decades and it works and it's effective. But where we really run into trouble is with males who are clinically what's called oligospermic. So what that means is that they produce very few numbers of sperm. So when you're producing very few numbers of sperm, whether in your ejaculate or if the patient is what we call azoospermic, which means no sperm in the ejaculate, then often surgical intervention to retrieve sperm, whether from the epididymis aspirates or whether it's from testicular tissue samples. And when you're working with that, it's very low numbers of sperm. It can be in patients. You're looking at single digits sometimes of live sperm. So you don't want to put them through that osmotic stress and 50% survival when you're working with very low numbers. So a newer technology that has emerged is something called vitrification. And vitrification is direct exposure of the sperm cells to the nitrogen, just directly from the room temperature to negative 196 C. So in the previous method that I had described, we expose the sperm to nitrogen vapors for a period of time, 15 to 30 minutes before we plunge them in the actual liquid. So it's a slow cooling rate that those cells undergo before the final frozen state. This is the exact opposite of that. This is taking it from room temperature and plunging it directly into nitrogen. And what that does, for anyone interested in physics out there, it transitions directly into the glass state.
Vitrification.
Yes.
Okay.
Glass vitrification. Yes, the same way glass is produced. Yes. So we do that with cells, again, to prevent ice crystal formation. And that has higher survival rate with few sperm cells. So that's a newer technology that's emerging. That's not offered in every clinic. In fact, there's very few that do offer it, but it's an emerging technology in the field.
So I have about 1 million questions, but what's the longest known sperm that have come out of freezing and been able to fertilize an egg?
Wonderful question. So at my facility, Reproductive Diagnostics, we've been in business for 35 years, and we still have samples stored from the beginning of that period. And those samples are still viable. So and this has been done, wide scale sperm cryopreservation has been around since at least the 1980s, late 70s.
Wow, so 50 years almost.
Yes,
Yeah.
getting there.
Yeah.
And as far as we know, there's no time limit to that because, as we talked earlier, they are suspended in time and there's no metabolic activity occurring in those cells.
They're all good. Yeah.
Yes. So how long the sperm samples will survive, as far as we know, indefinitely, but only time will tell at this point. Now, the oldest sample that has been used to produce a human offspring, it's over 25 years. That a sample has been frozen for 25 years and thawed and then used with in vitro fertilization to generate healthy human offspring embryos.
So on social media, we asked for audience input of what questions they wanted answered. And you've answered most of them, but there was one other one that didn't get answered. And a listener asked, how long will sperm last outside of the body after it's been ejaculated?
Interesting question, and we can go down several paths with that answer. So, one of the functions of semen is just to transport the sperm from the testes to the vagina to be deposited in the vagina for eventual escape through the cervix. And within the semen itself, semen is not a good preserver of sperm. So extended periods of time in semen, we see a linear decrease in sperm survival. Yes. So outside of the body, whether it's female body or male body, it'll last a few hours. Now, if we get that sample into a media in the laboratory, like we use for IVF, it can survive for a few days.
Just out on the lab bench, not frozen?
Just out on the bench, not frozen, or in an incubator in proper temperature, but not frozen.
Right.
Yes. And then within the female reproductive tract, they can survive in several days, 10 days even. So within semen itself, let's say on a surface, it's only going to last a couple of hours. In the laboratory, without being frozen, just in an incubator, we can make it last for a few days, 72 hours. And then within the female body, it lasts for days.
Well, thank you so much. Is there anything I didn't ask that I should have asked about sperm storage?
Instead of asking a question, a message that I want to get across is the importance of reproductive health. In summary, stay aware of your reproductive health and to not be afraid to ask questions or to seek counseling or even fertility preservation if you so desire.
Well, thank you so much for joining us, and I look forward to talking to you again soon.
Wonderful. Can't wait. Thank you for having me. It's been an absolute pleasure.
And thank you all for listening. Thanks also to our production team, Isabel Espinoza, Lane Patton, and Daphne Trilliana. The music used in this episode is from I Don't Smoke by the Mythical Score Society.